Michael, and so yes, it gives me tremendous pleasure to introduce Patrik Nosil today. I should say that I've known about Patrik's work for a very long time and admired it immensely, and so I've known about it actually for such a long time. I finally met him last year in Lisbon at the European Society of Evolutionary Biology meeting, and I was bowled over by how young you are and have achieved such an amazing work. But just to say what I just learned, I thought you were kidding there, Michael. So actually,
you might want to run and find him.
so Patrik actually originally came. He's a citizen of the world. So he came from way back when, from Croatia, and then did his work, his graduate training, his bachelor's work, all in Canada. So he graduated from the University of Victoria in '99, and then did his PhD with Bernie Presby at Simon Fraser University in Canada. From there, he went on to get a position at the University of Colorado in Boulder, and with working there, I think, just as an assistant professor. You didn't go you weren't associate there. You're too young. But then he was wooed away from Colorado to Europe. He got one of the most prestigious imaginable fellowships to go to the University of Sheffield in England, and where he currently is now. And so this fellowship will, which allows him to do almost what I learned, almost whatever he wants. And so Patrik's going to tell us about that, about all of the work that he's been doing today. I won't take up any more time except to say that he has earned an amazing number of awards and fellowships that I won't elaborate on. Besides
no
this.
One wants to hear.
Well, it is pretty outstanding. Besides this amazing European fellowship, he's also got awards, the Dubchansky Prize, a lot of NSERC fellowships, the Governor General Canada Gold Medal. It's really, well, it sounds just extraordinary. So thanks so much for coming. I should also say that for any climbers out there, Patrik is an amazing climber and incredibly enthusiastic about it, and
not as good as their son. But.
That's in large part why I could lure him to Berkeley, because there's a rock here that Patrik wanted. So thanks so much, Patrik.
So yeah, thanks for having me. I've been doing fieldwork now in California for, I think this is the 14th year, and I've actually never been to Berkeley, to campus or anything, so it's a pleasure to be here. Thanks for having me. And I'll be sharing some of this work that we've been doing, not quite in your own backyard, but all around California, and there are some populations and species of this system that live in the Bay Area, and there's some that are fairly close to here. But the kind of more general or central theme that will run throughout the talk, independent of the study system, is this issue of how repeatable and predictable is evolution. And I'd like to introduce this question with a thought experiment. It's a little bit cliche, but I think it gets us into the right mindset. This is a thought experiment proposed by Stephen Jay Gould in his 1989 book. Quoting from Gould, "I call this experiment replaying life's tape. You press the rewind button, and making sure you thoroughly erase everything that actually happened, go back to any time and place in the past, then let the tape run again and see if the repetition looks at all like the original." And then a few pages later, Gould concludes that if we ran this experiment, the repetition wouldn't look very similar at all if we replayed the tape of life. So again, quoting from Gould, any replay of the tape would lead evolution down a pathway radically different from the road actually taken. So in Gould's view, and in the view of some people, evolution is rather unrepeatable or unpredictable. So if we replay the tape of life, we would get very, very different outcomes, evolutionary outcomes, even under perhaps identical conditions. However, we know that evolution isn't always totally idiosyncratic. It can be predictable and repeatable. And of course, this stems from famous examples of parallel evolution or convergent evolution, where we see repeated evolution of the same phenotypic characteristics in different lineages, often in association with repeated colonization and adaptation to similar ecological habitats. So we know parallel evolution occurs. So what I'd like to do today is give this talk that has four main components. First, I'll give kind of a broader overview about my thinking about what we do know and what we don't know about parallel evolution and repeatability of evolution. I'll then introduce the stick insect system in California that we've been using to study repeated adaptation of different host plants and replicated instances of speciation. I'll describe an observational study on whole genome divergence between populations of these stick insects. And then I'll discuss a field transplant experiment that helps us validate some of the interpretations of the genomic data. So we know from a number of lines of evidence that evolution can be repeatable. So perhaps the most direct evidence comes from longer-term experimental evolution studies, for example, done by Lenski and many other colleagues, that have documented repeated parallel evolution at both the phenotypic level and at the genetic level. However, these instances of repeated evolution also have some effects of contingency and chance usually built in. So there's a nice example published in Science a few years ago by Justin Meyer and colleagues, where they had replicate lines of a phage co-evolving with E. coli, and these lines independently involve the exact same mechanism for attacking the bacteria. However, not every line evolved this adaptation, and every line that did evolve that adaptation used the exact same sequence of mutations. So there were some kind of constraints or contingencies built on top of these instances of replicate evolution. And a general theme emerging from this microbial literature is that evolution can be constrained during instances of parallel evolution. And this is because of this fact. Just because two mutations are beneficial on their own, it doesn't follow that they're beneficial together. So blue might increase fitness on its own, orange allele increases fitness on its own, put them together in a combination, and that's a deleterious combination that's selected against. And what this means is that this can constrain the number of paths that selection can take during adaptation, and these constraints should actually increase the repeatability or predictability of evolution. And I think perhaps one of the nicest examples of this is again a science paper now back in 2006 by Weinreich et al. And colleagues. And they studied this gene, the beta-lactamase gene, and it's known that there's 5 mutations in this gene that confer antibiotic resistance. So we have a wild type and a derived resistant type, and you need all 5 of these colored mutations here to get this critter that can resist our bugs. If you take these 2 different types, there's actually 32 allelic combinations that separate the wild type from the derived type. Some of those are depicted here. And more importantly, there's many mutational paths that separate them. So even for the exact same kind of movement from this genotype to genotype, one mutational path is to go to red, to orange, to green. Or exact same genotypes, another mutational path is first go to orange, then go to red, then go to green. So this would suggest, at face value, there might be a lot of ways to evolve antibiotic resistance, and evolution would be rather idiosyncratic or unrepeatable. And what they did in this paper is they created all of these 32 combinations, they competed them against one another in growth media, and they found that most of these combinations are actually deleterious. They reduce fitness below the wild type. And so they concluded, of these 122 possibilities, only 18 were really accessible to natural selection, and even these weren't equally accessible. So this suggested kind of constraints on evolution that would cause it to be repeatable. So if we move away from microbes, of course we know in nature there's evidence for repeatable evolution from replicate adaptive radiation. So certainly in this room everyone probably has their favorite replicate adaptive radiation. Here's one with cichlid fish from different lakes in Africa that have evolved convergently. We even at this point know a fair bit about the genetic basis of this type of convergent evolution, at least for instances of individual traits. There is a paper in Proceedings B by Gina Conti and colleagues 2 years ago now that did a literature survey or meta-analysis of types of how often parallel phenotypic evolution of a trait in different lineages involved the same genetic basis, the same genomic region. And it was quite often. So from genetic mapping studies, 32% of the time we saw reuse of the same genomic regions. From candidate genes, 50% of the time. So I'd say that this microbial work, the work on adaptive radiation, this literature survey suggests that there are these kind of constraints at adaptation which will drive evolution to be more repeatable and more predictable. However, most of our understanding right now comes from a focus on individual phenotypic traits or individual genes. So what I'd like to do in this talk is add some complexity to the situation, and in particular, two types of complexity. I'd like to consider the speciation process, which can be complex, and I'd like to consider genome-wide evolution, so move from a genic to a genomic perspective, and ask if we add this complexity, is evolution less repeatable? At the phenotypic level, at least, we know that even speciation can occur in replication, in replicate, and be repeatable. And this is this process of parallel speciation, coined by Schluter and Nagel in a 1995 paper in the American Naturalist. And really, all they did is extend this process of regular parallel evolution, which is depicted here, where instead of simply adapting to one type of environment, we have populations colonizing two different types of environments. So we're having kind of replicate parallel divergence. And the idea is that in correlation with these replicate ecological divergences, we repeatedly evolve reproductive isolation or barriers to interbreeding between these ecologically divergent populations. So in other words, reproductive isolation evolves multiple times and in correlation with these multiple ecological shifts. In '95, there were no conclusive examples of this, but since then, many have emerged. Perhaps the most famous one involves marine and freshwater stickleback, where the ancestral marine population has repeatedly colonized freshwater, adapted to freshwater, and as a consequence, evolved barriers to interbreeding with the ancestral marine population. We don't know very much about the genomic basis of this process of parallel speciation. So how repeatably do we see the same genomic regions involved in replicate instances of speciation? And before getting to the data part of the talk, I'd like to address this issue of, is a lack of repeatability kind of a straw man hypothesis? Do we already know that evolution will be quite repeatable even at the genome-wide level, given what we know about parallel evolution in microbes, at the phenotypic level, and adaptive radiations, etc.? But I'll argue that we might actually expect very idiosyncratic or non-repeatable evolution at the genomic level, and especially during the complex speciation process. And there's a number of reasons for this. I'll go over some. First of all, even if populations are adapting to similar ecological conditions independently, those ecological conditions are unlikely to be absolutely identical. So the traits and the ecological factors involved in different instances of speciation will vary, at least slightly. Even if the same traits are involved in those replicate instances of speciation, the genetic architecture of those traits could vary. So this will result in different genomic regions differentiating in different instances of speciation. Perhaps more so than at the phenotypic evolution, genome evolution is strongly affected by stochastic factors, such as genetic drift. And then finally, it's important to remember that even adaptive molecular evolution is affected by contingencies, such as the order in which mutations arise. So there's reasons to suspect evolution to be less repeatable at the genome-wide level and during speciation. So the actual data part of the talk that I'll talk about today is aimed at addressing these two basic questions. How repeated or parallel is divergence at the genomic level during parallel speciation? And what is natural selection's role in driving the repeated component of genomic divergence? So before going into any of the genomic data, I'd like to give background on the natural history and biology of this group of insects that I work on in California. So here's some beautiful photos of the stick insects taken by postdoc in the lab, Moritz Muschik. They're distributed really all around California, mostly kind of in the coast and a little bit east into the interior. And they're found on a wide range of different host plants, both angiosperm and gymnosperm host plants. As you can see, they tend to be adapted to the host plants they live on in terms of crypsis and camouflage. This is the ranges of the different species, so you can see us around here where you can go find stick insects if you know where to go look. But for today's talk, we'll focus here in an area in the Chaparral Hills around Santa Barbara, so about five hours south of here. And we'll focus on this species, Tabima christinae, shown sitting on one of its host plants. In those hills around Santa Barbara, it lives in these kind of bushy, shrubby, chaparral-type habitats, as depicted here. And most importantly for today's talk, it lives there primarily on 2 different host plants, and populations have adapted to these different host plants. So on top we have an individual which is adapted to the host plant Adenostoma. This is a small shrubby host plant in the rose family. And in the bottom here we have an individual adapted to the host plant Ceanothus, which can be a larger, more tree-like host plant. It's a buckthorn and it has broad, ovate leaves. They exhibit a number of differences, but perhaps the most conspicuous difference between the ecotypes is the presence or absence of this white dorsal stripe that's on the back. If you go out into the landscape, you sample stick insects from a population of Adenostoma, you'll see that 7 of the 10 individuals you capture on average they exhibit the presence of this stripe. In contrast, if you go and sample a patch of Ceanothus, on average only 2 of the 10 individuals will bear this stripe. These differences are heritable. We have these kind of frequency differences. They're generally not fixed differences between populations. Through a large number of past experiments, we've shown that these differences evolved at least in part due to natural selection from visual predators such as birds and lizards. So this is about as close as I'll come to vertebrate biology. In the talk, a student in the lab drew this scrub jay. They're hunting, and these different colors help them blend into the host plants on which they rest. So rather than showing the experimental data, I'll take a short break here and allow everyone to hunt for the striped morph, which is hiding on the background in which it is cryptic. So who sees it? You guys are better than the average audience. Okay, I'll give people a hint before I put. It's right in the center of the photo. Who sees it now? All right, well, you guys are actually way better than most audiences. So most people don't end up seeing it. So the stripe helps insects blend in against this background. It's actually conspicuous against the broad leaves of Ceanothus, and we have a number of actual experiments that demonstrate this. So populations have adapted to these different host plants. A major source of selection is bird predation. These differences have evolved repeatedly across the landscape. Populations grouped by geography, not by host plants. It appears there's been multiple shifts through this kind of mosaic network of the different host plants. During my PhD, what I wanted to do at the phenotypic level is test for this process of parallel speciation. As populations repeatedly adapt to these different host plants, do they repeatedly evolve barriers to interbreeding, reproductive isolation? And the short answer is yes. There's host-dependent reproductive isolation that exists independent from genetic distance. So if you take 2 populations adapted to the exact same host plant from different localities, and you measure reproductive isolation between them, we'll get into this in a minute, it tends to be weak or absent. In contrast, if you compare populations which are on different host plants, they tend to show moderate or stronger levels of reproductive isolation. And this is true for a number of barriers to interbreeding. First of all, they have slight differences in host preferences, such that they're somewhat hesitant to move between host plants at all behaviorally. If they do move to the other host plant, they're very likely to get eaten by a bird before they secure a mating. And a number of mating trials have shown that even if they do move between host plants, they're sexually mature, they find a mate, there's a reluctance for the different populations from different host plants to interbreed with one another. So there's pre-mating sexual isolation. And recent work in the lab by postdoc Rudy Reach is examining the role of cuticular hydrocarbons and smell in mediating this sexual isolation. An important aspect of this repeated evolution of reproductive isolation, though, is that it's partial, so it's not complete. Where the ecotypes co-occur in parapatry or sympatry, there's still some genetic exchange and they interbreed with one another. And what this means is that gene flow maintains a pool of standing variation within these populations. And this will be important for the experiment I'll talk about at the end of the talk. So to summarize the study system, we have these populations. They've adapted to these different host plants. They've done that in replicate in different geographical localities around Santa Barbara. And as a consequence of this repeated adaptation, they've repeatedly evolved some partial reproductive isolation. So now with the kind of background of the study system in place, we can turn to the question of how repeated is divergence at the genome-wide level during these instances of parallel phenotypic adaptation, phenotypic evolution. And so I'll start by describing an observational study quantifying whole genome divergence between replicate pairs of these ecotypes. So as with most genomic studies, there's a number of people involved. This is actually very short for a genome paper, but these are the different authors that are involved in this paper, which I'm lucky enough to say will be coming out in Science in a few weeks. And the important people, I'll highlight their exact role. A key person is the first author, Victor Soria Carrasco. He's a computational biologist, a postdoc in my lab in Sheffield, and he conducted the genome annotation, the functional annotation, some of the different tests for enrichment of specific types of genomic changes. So he did a lot of the computational biology. A number of other people are also involved in the computational end of things. The actual de novo assembly was done by Alex Berkeley. And then a lot of the more kind of population genetics, population biology aspects of the genomic analyses was led largely by Zach Gomper. And this is part of an ongoing collaboration with Jeff Feder, who I spent a year with at the Institute for Advanced Study in Berlin, Germany. And we first started kind of working on empirical and theoretical approaches to understanding genome variation. So we'll start with this observational study, and it'll focus on quantifying levels of genetic differentiation across the genome. So some regions of the genome might be relatively weakly differentiated, having low FST values, ranging through to other regions of the genome which are much more strongly differentiated, have high FST values. It's often assumed that these regions of high differentiation are affected by divergent natural selection. So divergent selection is pulling apart allele frequencies between populations, resulting in high FST. And this inference for divergent selection is bolstered if we see a region in parallel repeatedly showing high FST between multiple population pairs. So I won't go into the gory details, but I'll give you a little bit of background about what we did. The study starts, as with many genomic studies, with the generation of a reference genome. So with flow cytometry, we estimated the genome size. It's fairly large. For the bird people in the room, this is a bird-sized genome, 1.3 gigabases. We conducted sequencing of libraries of various insert sizes and assembled those into about 14,000 scaffolds, and then conducted gene, functional, and transposable annotation, element annotation of these scaffolds. This resulted in about 16,000 genes with a predicted function, 4% of the genome being classified as coding, 19% genic non-coding, and 77 intergenic. So now we have these 14,000 or so scaffolds. We have them annotated, so we have an annotated genome. But we wanted to get a better handle of the organization of these scaffolds across chromosomes. So we conduct genetic crosses. In this particular instance, we had 3 mapping families that yielded 186 offspring. We sequenced these using more of a genotype by sequencing type methodology, which resulted in just over 21,000 recombination informative SNPs. So this allowed us to estimate rates of recombination between and within scaffolds and assemble these 14,000 scaffolds into the putative 13 linkage groups or 13 chromosomes that this species exhibits. So with this annotated genome assembled into linkage groups, we turn to what to me is the interesting part, and that's the population biology. So we conducted whole genome resequencing of 160 individuals from 4 replicate ecotype pairs. So we have 3 ecotype pairs in 3 different localities where they occur together in geographical parapatry, and then we had 1 ecotype pair where they're separated by about 6 kilometers or 4 miles. This whole genome resequencing yielded about 4.5 million SNPs, and this is what we've subjected to further analysis. So there's 3 aspects of analysis I'll go through: population structure, variation and divergence across the genome, and then how parallel was divergence across these 4 different pairs of ecotypes. The population structure simply kind of confirms what we already knew, and that's that populations tend to group by geography, not by host plants. So the host plants are mixed up at the tips of their phylogeny, and we have strong grouping by geography. And this is supported by any type of analysis of structure that we do. More interestingly, we turn to variable divergence across the genome. So this is also actually expected. We see highly variable divergence across the genome, but I'd like to point out a few points. First of all, the level of differentiation we're looking at here is on average rather low. So the median FST is 0.087. This is kind of the initiation of adaptation and speciation, if you will. And then the range is quite large. We have many undifferentiated loci, and then FST ranges up to 0.8. So to depict that graphically, we have the mean down here, but the maximum is actually quite high, and we observe no fixed differences whatsoever. So what we wanted to do is quantify how this variation in differentiation was distributed across the genome. So another way of thinking about this is look for whether we have peaks or islands of differentiation in the genome, and ask how many of them are there, how large are they in size, and are they clustered or are they distributed all over the genome. So the challenge here comes in finding an objective way to quantify this variation, because each SNP is kind of just jumping up and down in its level of differentiation. This is the raw data where we have our 4 replicate ecotype pairs. Each points a different SNP, and there's variation all over the place. So we wanted to quantify this. What we chose to do in the end is adopt this approach, which was put forth by Laurent Escoffier and colleagues in a paper in BMC Genomics recently, and they applied this to human data. And it's a hidden Markov model approach, which essentially what it does is delimit runs of low differentiation that are separated then from a run of high differentiation followed by another run of low differentiation. So it looks for these kind of runs of homogeneously low or homogeneously high differentiation, but allows individual SNPs within these regions to not necessarily be highly or lowly differentiated. So it's looking for these kind of clumps of differentiation in the genome. This is kind of a stylized, this is real data, but this is one of the genomic islands that looks like a stylized island. The real model outcome looks a little bit more like this, where you have the peaks of divergence are more differentiated, but often not radically so. And this is the zoomed out view of the whole process. When we run this across the genome, what we find are thousands of small to modest sized regions of divergence in the genome. So in number, they range across the ecotype pairs from 7,000 to 18,000. Their size is rather small, 2 to 5,000 base pairs, and they occupy somewhere 15 to 20% of the genome. So there's many kind of small regions of differentiation in the genome. But many small regions of differentiation. But now to focus back on the main topic of the talk is how replicated or repeated are these regions of divergence in the 4 ecotype pairs. So if we have one of these peaks of differentiation in one of our ecotype pairs, is it also represented in a second ecotype pair, and as a peak in a third ecotype pair, and as a peak in the 4th ecotype pair? So we did a number of different analyses, both for these hidden Markov model regions as well as for individual SNPs. They gave really congruent results. So explain the results for individual SNPs here today. Bottom line is that most of the divergence was unique to individual population pairs. So there's a lot of non-repeated divergence. So for example, if you take the FST distribution, you take SNPs that are in the tails of that distribution, for example, in the top 10% of the FST distribution, and if you ask if a SNP is in that tail of the distribution in one population pair, how often is it in the tails of the distribution in other ecotype pairs? And the answer is not very often. So 83% of the time, a SNP in the tail of the FST distribution was so in only a single ecotype pair, largely non-parallel divergence. However, we do see some parallel differentiation. So 17% of the time, for example, if a SNP was highly differentiated in one population pair, it also wasn't in another population pair, or even in all 4 population pairs. And with various types of permutation tests, we can show that this level of parallelism is actually more than you expect by chance. So in an absolute sense, it's not maybe a ton, but it's more than the null expectation. So for example, with permutation, you would expect about 400 SNPs just by chance to have high FST in all 4 of our replicate ecotype pairs, and we see about twice as many SNPs that actually show high FST in all 4 of our ecotype pairs. So we do have parallel divergence and more than expected by chance. Another aspect which I won't dwell upon, but in case anyone is interested in functional genomics, is we can ask what types of changes are involved in this parallel divergence. First of all, in these regions of parallel differentiation, there's an excess of coding changes. So there's about 1.5 times more coding changes in these parallel regions than expected by chance. There's also an overrepresentation or enrichment of specific molecular functions. For example, we'd expect only 7% of the SNPs in the genome to be associated with this function of metal ion binding, but in these peaks of parallel differentiation, almost half of the SNPs are assigned this molecular function. So to summarize the observational part, we see population structure grouping these populations by geography, not host. We see thousands of modest-sized regions of differentiation scattered across the genome. Most of this divergence is non-parallel. So most regions of divergence are very unique to an individual ecotype pair. But we do have parallelism, 17%, if you want to hold on to that number. And there's more parallelism than you expect by chance. And this parallelism is associated with an excess of coding regions. So that answers the first question, how repeatable is parallel divergence at the genomic level? We can say it's 17% repeatable. The next question that I'll conclude with is, what is natural selection's role in creating this repeated component of genomic divergence? So I alluded to earlier that often loci with high FST values are assumed to be under divergent selection, and if we see the same region repeatedly showing high FST, this kind of bolsters, by inference, the evidence for natural selection. However, the situation, as many of us know, is much more complicated than this. Patterns of genomic divergence are affected by many processes other than natural selection. So in addition to selection, rates of recombination, rates of migration, effective population sizes, mutation rates, etc., they will all affect genome variation and thus levels of genetic differentiation. Thus, inferring process from pattern can be difficult, which is why I put a purposely kind of blurry image here of FST across the genome. And in fact, these different processes can even be involved in scenarios that will result in parallel regions of high divergence, even in the absence of natural selection. So you can imagine a scenario where you had allopatric populations. They differentiate across a lot of their genome in allopatry, let's say purely by drift, just a thought experiment. They come into contact, they start interbreeding with one another. The regions of low recombination will maintain their differentiation much longer than the regions of high recombination. And so you could end up with this type of a pattern, even in parallel, with no role for natural selection. So we would like to have some kind of validation or test whether natural selection created these 17% parallel regions of divergence in the genome. So I'll describe at the end here an experiment that was designed to do this. So the hypothesis that this experiment tests is that repeated genomic divergence is driven by parallel divergent selection between host plants. And the prediction here is tailored a little bit more to the system and the experiment we did. And the prediction more specifically is that these regions of parallel genomic divergence between these natural population pairs, where we sequence their whole genomes, they will be enriched for SNPs that repeatedly show allele frequency divergence between host plants in field transplants. So we should see selection responses in actual transplants that map to and match differences in nature. So to test this hypothesis and these predictions, we conducted a classical type of ecological experiment, a transplant experiment. It starts by going to a large variable population of the host plant Adenostoma. From this, we collect over 2,000 individuals. 31 of those we keep immediately. We put them into ethanol. We ended up sequencing them using a genotyping by sequencing technology. And what this gives us is a representation or an estimate of the ancestral allele frequencies before we executed our experiment. We then take another 2,000 individuals and we transplant them into the wild, into a slightly different location in a paired blocks design. So the experiment has 5 blocks. Each block has a plant individual of the host plant Ceanothus and a plant individual of the host plant Adenostoma. So with 2,000 individuals transplanted, this means we're putting 200 onto each individual bush. The way the natural history of these bugs work is we transplant fairly young bugs. They kind of grow through the spring into adults, then they're mating as adults and laying eggs. By end of May, beginning of June, they've laid their eggs, and the eggs undergo a big diapause. Over the entire summer and winter, they're just in the soil as eggs, and they hatch again as the next generation the following spring. So after transplantation, we basically do nothing. We wait a year. We come back 1 year later, so this is exactly 1 generation later, and we come and collect any bugs that are on these experimental bushes, and these represent the F1 offspring of the transplanted individuals. We then collect genomic data from these F1 offspring as well, again using a genotyping by sequencing type of approach. And what this allows us to do is estimate allele frequency changes across a generation at about 82,000 SNPs. And what we're interested in specifically is not just whether allele frequencies changed, but whether they diverge between host plants and whether they diverge between host plants in parallel. So really what we want to do is calculate this kind of parallel divergence between host plants. To what extent at each SNP do we see an allele consistently going up on one host plant and consistently going down on another host plant. So of course many regions of the genome won't do this very much. Some will do it more, some will do it less. So we calculate this parallel divergence between hosts for each of our 86,000 SNPs. This is a composite quantile plot that shows the results. So you can see most regions of the genome show not very much parallel divergence between hosts, as you might expect. However, there is this kind of longer tail here, regions of the genome that showed more pronounced and consistent divergence between the host plants over the course of this experiment. Another way of looking at it is this. So this is averaged across the 5 different blocks. It just shows that for these SNPs in the tails of this distribution, if an allele tended to go up on one host plant, it tended to go down on the other host plant. So parallel divergence between host plants. So the key question is, and this is what I'll kind of move towards as I close here, is do these regions of the genome that are consistently diverging between host plants when we conduct this transplant, do they correspond to or map to the regions of the genome that are differentiated in our natural ecotype pairs that we just sequenced the natural population pairs? And more specifically, do these regions that are consistently diverging between host plants in our experiment, do they map to the regions which are not only different in nature, but that are different in parallel and replicate between different ecotype pairs? So this is the main kind of experimental validation or test of whether these regions of parallel genomic divergence were created by host-related natural selection. And the answer is yes, they are. The SNP showing the strongest experimental change between host plants in our experiment were in these regions of parallel divergence between host plants more often than expected by chance. So not massively more often, and it's interesting to think about why, but more often than expected by chance. So with randomization, again, we can show we'd expect about 20 SNPs that are in the tails of this distribution to map to regions of parallel high differentiation, and we see 32 that do so. So if we come back to our initial analogy of how repeatable and predictable is evolution, as with most things in biology, it's a quantitative question. Of course, Gould's partly right and partly wrong. But at least we can start putting some numbers on this with the stick insect system. So stick insect populations are examples of parallel speciation. They exhibit thousands of modest-sized regions of differentiation across their genome. This divergence at the genomic level is largely non-repeated across population pairs. So there's a lot of idiosyncratic or unique differentiation. However, there is a repeated component. It's more than expected by chance, 17%. And this repeated component is enriched for both coding genes, and it's enriched for these selection responses in this field transplant experiment. So if you were going back to Gould's thought experiment, I'd say that in the stick insect system, at least, these ecotype pairs, the answer was that if one were to replay the tape of life, it would likely result in many idiosyncratic outcomes. But even at the genome-wide level, and even during the complex speciation process, there would be a repeated component, and this repeated component would be driven by divergent natural selection. So that's all in terms of actual data, and so what I'll use with the last few minutes here is just to explore a little bit about where I'm going next and what some of the interesting remaining questions are. So when I introduced the biology of the system, I pointed out that reproductive isolation is only partial between these ecotypes. So gene flow between them is still rather high, linkage disequilibrium in the genome is low, genetic divergence is weak, etc. So they've diverged into what we generally call our ecotypes. They've evolved some reproductive isolation, but they haven't split into what would be considered distinct species under any species definition really that I know of. So the question is, one, why one species and not two? Unfortunately, we don't live long enough to just sit around studying these exact ecotypes for another 10,000 years to try to figure out this question. But we can take kind of a retrospective, indirect approach. And that's using closely related groups of stick insects in California that have diverged further down that continuum of speciation than these ecotypes around Santa Barbara. So there's 20 some odd species across California, and they exhibit a wide range of phenotypic and ecological differentiation. So instead of being on two angiosperm hosts, we have species pairs and even populations within species where one's on an angiosperm, one's on a gymnosperm. And we have all kinds of morphological diversity in terms of coloration. And they also exhibit a lot of elevational differentiation. So right now what we're doing is quantifying a lot of this kind of variation in divergence. And you can see that you have entities that are very differentiated, look like biological species. Then you have kind of more mixed populations and things that are uniform. And ideally what we'd like to do is replicate some of these studies that we've been doing with these bugs around Santa Barbara with taxa that have diverged a little bit further and then even maybe even near to the end of that continuum of speciation. Another aspect that I won't actually present any results about, but I'd like to point out is that with several colleagues, I've also been involved in a body of formal theoretical modeling work about the processes that drive genomic divergence and create different patterns of variation in genomes. So if anyone I talk to, I'd be happy to talk about the more theoretical aspects of this work as well. So to conclude, I'd like to thank some of the other main people involved. Aaron Camo here is visiting, considering maybe trying to do a postdoc at Berkeley. He's involved in mapping the genetic basis of many of the phenotypic traits that separate these ecotypes, as well as understanding how selection acts on those different phenotypes and what the genomic consequences of that are. There's another kind of backstory that I didn't talk about today about how evolution within these stick insects actually has knock-on effects on entire arthropod communities, because birds show up, eat the stick insects where they're not well camouflaged, and then eat the other spiders and caterpillars and beetles that are there. Timothy Farkas is leading that. I've acknowledged Victor already as really being involved in all the computational work. And the work examining the kind of variation across the speciation continuum is being led by another postdoc in the lab, Moritz Mushik. As I alluded to earlier, most of this kind of thinking, I worked on speciation from really much more of a phenotypic angle until about 2008, 2009, where I spent this year in Berlin as part of a working group on speciation with gene flow organized by Axel Meyer. And there, particularly, Jeff and I started collaborating together a lot, so I'd like to acknowledge his role in ongoing collaboration there. A number of other people, and of course, especially thank the European Research Council who funds this work. So thanks again for having me.